Remote users, can you hear me? Okay. I'm sharing my slides. Can you see it? Okay. So you may be a little distracted. Okay. I still have to continue with lecture, so please do your best to try to focus. And if you are not able to or zone me out for some parts of the lecture, then you remember you have that pre-recorded video, so please go ahead and watch it. Unfortunately, today I have to talk about humoral immunity, and one of the concepts there is related to maturation of lymphocytes that occurs in the red bone marrow and the thymus. And that's one that even with your fullest attention, I still lose students, right? So yeah, it's one of those days. I'm going to do my best to explain it, and hopefully you'll follow along. And if not, you got the video, and maybe we'll have time to come back and repeat it next class. Okay. All right. So before I go into lymphocytes, so there's a few things I want to complete talking about related to innate immunity. So we need to wrap up inflammation and then talk about complement activation as well. So let me talk a little bit about this. Inflammatory response is very, very important. It is part of the innate mechanism. Innate mechanisms, we said, were non-specific, which means we're not going after one very specific target. It's you don't have a receptor that recognizes one specific target. It's just basically any foreign target, right? So your non-specific phagocytic cells would be neutrophils and macrophages, and they basically are looking for any foreign or non-self antigen, and they know what to do with it, right? So again, macrophages and neutrophils will basically go through this process called phagocytosis, where they're internalizing the foreign pathogen, and then it binds with lysosomes, where you have very, very caustic, low pH, acidic, hydrolytic enzymes that can basically degrade that target. Neutrophils are your first responders, typically within about 8 to 10 hours or so. You'll see a peak response about 10 to 12 hours. And then basically they also kind of die in the process. So yes, there's a constant turnaround, right, of those neutrophils being produced back in the red bone marrow. Macrophages really differentiated from monocyte cells, and monocytes work very similarly, except they don't really eliminate themselves in the process, right? So the other important piece related to innate immunity is inflammation. All right, so inflammation is always a scary thing when you talk about it. Okay, it's not necessarily a bad thing. In an acute setting, it is a very good thing. It's a very essential thing that needs to happen to clear up the infection or to fight, to allow for resolution of injury, healing, and all of that. So, but again, it needs to be controlled in an acute setting. The when it becomes a problem is when you have chronic inflammation. So that's when it's uncontrolled, unregulated, and then you have an unnecessary, exaggerated response, right? But again, in an acute setting, very important. I need it, right? This needs to happen in order to recruit many other cells and to kind of bring together the different arms of the innate and the adaptive response. So let's talk a little bit about this. So the very first thing that happens during inflammation is that you're really sounding an alarm, right? This is what we call a chemical alarm. A whole bunch of different pro-inflammatory chemicals and molecules and mediators are released, okay? And a lot of this is released by those basophil cells. You remember basophils, right? They were packed with granules that contained histamine, and some other immune cells which act very much like basophils called mast cells. They also have histamine. That's the first thing that happens is like you have like a flooding of histamine, right? I mean, that's the first thing that kind of happens. Then as you recruit more and more cells, like macrophages and other epithelial cells that are kind of all responding to this injury or this inflammation situation, well, then you have what I call I don't know if you see it here, but it's kind of called the cytokine storm, right? So it's basically where you have a lot of other pro-inflammatory mediators called cytokines. That's just one class though that gets released. And so some of these mediators are again histamine, kinins, prostaglandins, and I'm going to talk to you all about complement proteins as well. So that all of this kind of sounds the alarm. This kind of sets the stage. All right. So what does this do? So all of these inflammatory mediators, like histamine, for example, will do the following things. I have a schematic coming up on the next slide, but we'll piece all of this together. And I'll tell you what happens to the arterioles. And then, of course, as you go further and further, the exchange vessels, the capillaries, what's going on there, right? But ultimately, let me back up a slide a second. Yeah, okay. The all of those effects that you see would result in these cardinal signs that are related to acute inflammation. You're going to see redness, heat, swelling, which is edema. You're going to see pain, right? And then, of course, all of this will hopefully improve or to resolve this injury or inflammation. Okay. All right, so here's the schematic that brings this entire inflammation concept together. So for example, let's say you have an injury right there, you cut your hand, right? I mean, so of course the clotting process is also going to get activated. We talked about this with blood. So this is the example of, okay, clotting being part of the resolution to injury, but this doesn't have to be, a wound or anything like that, right? It could be infiltration from a pathogen, and therefore you're just mounting an immune response and therefore inflammation related to it. So we'll go with this example. So here's what's going to happen. When you have injury, you have acute inflammation that sets the stage for a lot of different things that need to happen that allows for resolution of that injury. So ultimately, what you want to do is to resolve that injury, and this is healing. Good. Okay, we good? That's fine. Sorry about that. Um, Temperature seems to be a big issue in our building yesterday and today, especially like, I mean, like there are some labs that are at 48 degrees and 50 degrees is extremely brutally cold. So that was the interruption there. I'm sorry. Have to cancel a few labs this morning. But I think A&P, we're good. And those of you taking exams, you all are going to be in the computer room. I'm going to say we are okay in that room. We are better than 48 degrees. Okay. I think we're okay. All right. Sorry about that. Um, all right. So when you have an injury like this, right? So there's different things that are occurring, and I'm going to break this down for you. I'm going to start with the left and work my way towards the right. Okay. So when you first have tissue injury, like we said, we have that alarm, right? Or that chemical alarm going off where you have all these different inflammatory chemicals being released, like histamine, complement, and so on and so forth. And what does this do, right? So this is going to bring about a few changes in the blood vessels leading into that injured site. So you all remember how you have the arterial side that's carrying oxygenated blood to a tissue bed. So if you have injury within a tissue bed, the larger arteries, which of course are your arterioles leading into that tissue bed, what happens there? These inflammatory mediators cause dilation of those arterioles, right? Remember, arterioles are resistance vessels, so we can change the smooth muscle contraction, so on and so forth, allowing for widening of the diameter or constriction. In this case, what you're seeing here is inflammation actually promotes vasodilation. It opens up the lumen. So which means, all right, so then you're actually allowing more blood to flow into this injured site, right? And that's kind of counteractive if you kind of think about it, because if you're trying to stem blood loss, right, especially in this situation where you're talking about clotting and everything that needs to happen, well, you're actually opening up the blood vessels there. But this is actually an inflammation response, right? But of course, what's being brought in to that injured site? If you're allowing more blood there, then the blood's carrying something, and you know it's carrying clotting factors, it's carrying other things that are going to allow for resolution of that injury. So what you're seeing here is an increased blood flow to this inflamed site. This is called local hyperemia, where you are allowing increased blood supply to that region. And this is why you see these two uh, characteristic signs, right? So heat, again, inflammation, typically when you touch an area that's inflamed, it's kind of, first of all, it's red, right? And it also is kind of warm, kind of warmer than the rest of your body. So both of those are related to increased blood flow into that specific inflamed area, right? Okay. Um, Also, then, as you increase blood flow into that specific tissue bed, then what happens to the capillaries? Because the capillaries are the exchange vessels inside of those tissues, right? You're going to see increased permeability of those capillaries. So what that means is that basically you're going to be able to have more gaps between those endothelial cells, increasing the permeability for whatever is flowing through those capillaries to be able to exit those capillaries, right? And what's really happening here is, of course, you want clotting factors and things like that to be released from the blood so that you can resolve injuries like that where you have a cut, right? You have to actually go through that clotting cascade. But you're also bringing in a lot of other things in that blood. Like, for example, when you sound the alarm and you got that chemical alarm going off, back in the red bone marrow where you have your stem cells, the hematopoietic stem cells, they're responding to these signals. And now we know, okay, we've got to increase our WBC numbers. So remember we talked about that, right? Leukopoiesis, right? So then what are we increasing? We're probably increasing neutrophil numbers. We're increasing monocyte numbers. We're probably also increasing lymphocyte numbers, right? So all of these cells are increasing in numbers, and you have leaky capillaries, right, that are allowing these cells to leave those blood vessels so that they can get to that site of injury, right? So all of this is helping with resolution of this injury. So if I didn't have that inflammation, all of these processes would be obviously a lot more sluggish and not so robust, right? We definitely want this. Now, as the capillaries leak, well, as they become more leaky, you're also losing plasma, right? You're losing fluid, and that's what's causing the swelling, the edema. And as the fluid accumulates in those interstitial spaces, there's nerve endings in those areas, and you're pushing up against receptors, nerve endings, and that's causing the pain, right? So those are all cardinal features related to inflammation, okay? So I want to talk a little bit more about how we're recruiting more WBCs, right? So different varieties of WBCs and what is it going to do? So as we're resolving the injury, we've got clotting factors that's triggering the whole clotting cascade, helping with that fibrin mesh and all of that that we talked about with the blood chapter. At the same time, we are also attracting more neutrophils, monocytes, lymphocytes, all of that to this inflamed, injured area, right? So this is going to tie into this last piece here, which is what we call phagocytic mobilization. So it's shown here in a flow chart, but actually there's a schematic coming up where I can kind of piece this together. Phagocytic mobilization. So again, when you're thinking about phagocytic cells, this is neutrophils and the macrophages. Your first responders are neutrophils, and then right on their heels, you got monocytes that would arrive, then transforming into macrophage cells. Once these two are mobilized, we're going to talk about how that happens and then, of course, resolution of that injury, right? The different steps here are shown here. So the first part is leukocytosis. This, again, is not, this is something we talked about in the blood chapter, where, again, in response to different like colony stimulating factors, certain inflammatory mediators like interleukins and so on and so forth, which is, you see, there's overlap with the inflammation process here, right? Those are my triggers to tell those hematopoietic stem cells to increase neutrophil numbers, to increase monocyte numbers, and so on and so forth. That's the first part, leukocytosis. Then as they leave the red bone marrow and enter the blood, then they get to this site of injury. This is where we need to talk about what needs to happen next, right? So those are the next three steps here. I'm going to use my schematic to explain it. All right. I have an injured area right there. I have to, so you're going to see inflammation, acute inflammation occurring. That's going to help with resolution of injury. So if this is my injured area, all right, let me get my pointer to work. Here's the capillary feeding into that particular tissue bed, right? So I've got leukocytosis occurring in the red bone marrow, and you're seeing neutrophils here in this blood vessel. So I have elevated neutrophil numbers happening very early on. I don't know, like the first, like I said, 8 to 10 hours, right, of that injury process. So that's the first thing. Leukocytosis allows those cells to arrive somewhere close to the injury area. Now, those capillaries are very leaky, right? And of course, the histamine and things like that, prostaglandins did that, allowed it to be leaky. So the next thing is margination. These neutrophils or WBCs of any sort, they are going to basically flatten themselves against the wall of this capillary. That's called margination. They're kind of clinging to the capillary wall, right? Then they're going to squeeze out in those spaces between adjacent endothelial cells. This is called diapedesis, right? And once it leaves the blood vessel, now you are in the general vicinity of where that injury has occurred, but I may not exactly be at the injury site. So the last part is actually chemotaxis. This is where, again, all of these injured cells here are releasing those cytokines, all of those different inflammatory mediators, right? So this is what I call the chemicals. These chemicals, so these neutrophils, for example, they literally sniff out those chemicals. They follow a trail, right? A chemical trail to get to that exact site where they're needed. Now, if you have a site of injury there, maybe those green structures there might be bacteria, might be a virus, maybe whatever it is. You have infiltration of a pathogen from the outside. So what would those neutrophils do? They're going to engulf that bacteria into the neutrophil, and this is phagocytosis, and then it binds with the lysosomes, and you would eliminate that target. Except, like I said, the neutrophils also die in that process. That's why they have a very short life. But instead of a neutrophil, if that was a macrophage, it would do the same thing, except it's not going to kill itself in the process. Right? Okay, that's it. So we talked about how inflammation, you got that chemical storm that's being created, and that's going to allow several different things to occur. The arterioles, which lead into a tissue bed, they dilate, which means you have increased blood, local hyperemia, right? Increased blood flowing into that region, and that's what's causing that redness and so on and so forth. Then you have the capillaries that become very leaky. That's going to allow for this whole phagocytic margination process to occur, right? And of course, along with the cells, you also have clotting factors and other things that need to resolve the injury. All right, so that's the entire inflammation process. So we're going to move on, talk a little bit about interferons next, and then complement proteins. So interferons are mostly molecules or factors, proteins really, that would help with resolution in the case of a virus. So it's an antiviral, really. So I'm going to show you what this looks like. Oops, went too fast. I have two cells here, right? Two cells adjacent to each other. The cell here on the left is the cell that's being infected by the virus. So I have the plasma membrane. Purple portion is the nucleus with the DNA inside of it. And so I have this virus that breaches the plasma membrane, and then notice it enters through those nuclear pores. It actually infiltrates the nucleus of this host cell, right? So the way that the virus works is that it does not have the right machinery to replicate itself, to make multiple copies of itself. So how it does this is it takes over, it hijacks the DNA of this host cell, and it redirects the DNA, which has like, you remember the DNA has like gene sequences, which then, from that DNA, you can make mRNA, and the message can then be translated into a protein, right? So it kind of hijacks certain portions of the DNA called promoter sequences, and basically it redirects that particular gene to not make whatever protein it's supposed to be making, but to make more copies of the virus. Does that make sense? Right? So this is basically what we call viral replication. So this cell, it has been infected, and it's obviously making all of these copies of the virus. Now, there's not a whole lot of hope for this infected cell. This cell is now needing to be eliminated. Hopefully, we're going to mount an immune response. It's going to take care of that infected cell. But this infected cell, this virus infected cell, can sound the alarm and protect the neighboring cells, right? So this is what's going to happen. When the virus infects a cell, this infected cell also starts producing something called interferon proteins. And there's a whole lot of varieties of interferons. We'll just go with interferons, all right? So what's going on here is these red triangles are these interferon proteins that are being produced by the infected cell. And you're seeing exocytosis of those interferon proteins, which then alerts this neighboring cell, which is not yet infected, right? So there are receptors, interferon receptors on these cells that are binding these proteins. And that's basically alerting the cell saying, we have a virus in the vicinity. It kind of looks like this, right? So what's going to happen? These interferon proteins are going to direct the nucleus of this neighboring cell that's not yet infected to make antiviral mRNA. Does that make sense? Which makes antiviral proteins. And now as the virus tries to enter the cell, it's blocked because you have antibodies that already recognize that virus and say, no, you cannot enter. Does that make sense? So we're preventing this cell, I'm sorry, the virus from infecting a neighboring cell. That's how interferons work, okay, which is an antiviral. Okay. And then the last piece is the complement system. So this is very, I mean, it has a lot of different factors, a group of proteins, very similar to like your clotting factors, right? I mean, so they kind of all work in, they have to kind of aggregate together and form complexes. So I won't go into all the specifics, but I do want to show you how it overall works, okay? So you have different types of blood proteins, about 20 of them, that make up the complement system. So I'm going to show you how we activate these complement proteins, because that's very important for what we need to talk about next with the lymphocytes, okay? But once we activate these proteins, I'm going to show you that how a target cell can actually be eliminated by these complement proteins. That's one way to do it. There are a few other ways. So let's talk about it. Words like explaining the whole activation process and the whole cell lysis, and I'll talk to you about membrane attack complex and all of that. Let's actually go to our schematic. All right. So there's three different ways by which we can activate our complement proteins. So complement proteins are basically just like clotting factors. They are like kind of inactive, right? I mean, we don't really need it to be activated unless they're necessary. So this is what happens is antibodies from your lymphocytes, B cells, can actually allow for activation of complement proteins. I want to go ahead and start mapping this out a little bit. I'm going to show you this in a little bit, but I'm going to start here. If I have a B lymphocyte, right, this B lymphocyte is programmed to identify one specific foreign antigen because it has a very specific receptor, BCR, B cell receptor, okay? When that receptor identifies a foreign target, and I'm going to go with, here's my foreign target, this is some kind of a virus or whatever, right? When this B cell receptor says, yeah, I recognize this particular foreign pathogen, right? It's going to bind that foreign pathogen or fragments of it or whatever. And basically this foreign antigen plus the receptor, the complex that's formed, is going to activate that B cell. I'll add more details. But then that B cell, okay, it was initially a naive B cell, meaning it had the receptor but had not yet encountered that foreign pathogen. But now that it has encountered the pathogen that it's supposed to do something about, now we've activated this cell, right? So when we activate the cell, this becomes a specialized cell and it differentiates. It's no longer a naive B cell, right? Naive is I've not really encountered the foreign pathogen. I know what I'm looking for, but I haven't encountered it. But once I've encountered that foreign pathogen, this is no longer naive. It's an activated cell. It differentiates into something called plasma cells, right? And these plasma cells then start secreting antibodies. Antibodies are proteins, okay? These antibodies look very much like that receptor, right? It's a whole bunch of these receptors. Well, antibodies are proteins that then circulate in the blood and lymph, right? So now, wherever these plasma cells are getting activated, you all remember the lymph node we talked about last class, right? So in the outer cortex region where we talked about, okay, I'm certainly not even in view. Hold on a minute. There we go. Um, in the follicle region where you had B cells, that's really where they could maybe encounter that foreign pathogen, where they get activated, and you see proliferation occurring, where they become those plasma cells. All right. So these plasma cells may be in the lymph node, somewhere in the lymph node, right? Maybe the neck region, right? But then once those antibodies are released from here, now we're going places. This is systemically distributed, which means that these antibodies can now recognize that virus anywhere in the body. Does that make sense to you? Yeah. So what happens? These antibodies are going to find that virus wherever it is in the body, and it's going to tag it on the surface. You see it? That's called opsonization. Okay, that's the way that those antibodies are going to work. Of course, there's a lot more details. I'll come back and fill that in. But for right now, for complement proteins, okay, how do we activate complement proteins? If we recognize an opsonized target like this, where you have something foreign that's been coded by these antibodies, that could activate the complement system. That's your classical pathway. Does that make sense? This is saying, here's a target cell, this virus that needs to be eliminated. And how do I know it's a target cell? Because the surface of that target cell is coded, you got markers on it, which are your antibodies, right? Okay. Sometimes there's also other proteins like lectins that would get activated, and the activation of those lectins would cause the activation of your complement proteins as well, right? And the last part is an alternative pathway. It's kind of like a spontaneous activation. So basically, every cell that belongs in your body, correct, has inhibitors on its surface that prevents unnecessary activation of the complement protein, right? So, okay, let's do this. If I had a normal kidney cell here, right? This is normal. I've got inhibitors on this kidney cell that prevents complement proteins to be activated. Because if complement proteins got activated, I would then eliminate this normal kidney cell. Does that make sense to you? Which I don't want, right? But if I had a foreign pathogen like that, they don't have those inhibitors on their surface, right? So the absence of those inhibitors can spontaneously activate those complement proteins, making it a target for elimination. So there's 3 different ways we can activate our complement proteins. Classical pathway is the presence of antibodies, opsonizing, right, the surface of that foreign target. Lectins can do the trick, but also spontaneously, because you don't have the inhibition to the complement proteins, that can cause activation. Regardless of how that happened, right? When you activate the complement protein, this is what's going to happen. So you don't need to know the details, but you can see here that several of these blood proteins come together to form like a complex. They have different roles, so we'll discuss this. C3, for example, can further opsonize the target, right? So, I mean, okay, so maybe if a target was, if complement proteins were activated by the presence of these lectins or the alternative pathway, now the complement proteins are also opsonizing the target. So just like those antibodies, maybe we've got complement proteins that are also coating the surface, saying opsonizing that target, right? So that's one way of doing it. What else can the complement network of proteins do? They can enhance inflammation. Everything we just talked about inflammation, that can help with, mostly it's the histamine release, right? So therefore, what does it do to arterioles? Dilation, and then making those capillaries leaky, allowing for phagocytic cells to be recruited, all of that. It helps with that. It enhances inflammation. But this part's interesting. This is where you have several different proteins coming together, forming a complex, C3, 5, 7, 6, all of that, right? So don't need to know the details, but you're forming a complex, and this is called MAC, membrane attack complex, MAC. Okay, here's what I'm going to show you. Imagine, you'll recognize the phospholipid bilayer, correct, right? I mean, so imagine that this was a plasma membrane of a target cell, right? What if that kidney cell there got infected with a virus? What if this became a cancerous cell, right? So that's a cell that I want to now eliminate. So what happens when you have this complex formed? All of these proteins come together and they form a membrane attack complex, and it's basically a channel, right? It's a channel that inserts itself in the target cell's plasma membrane. If you have a channel, something can come in or leave, right? So what's going to come into this target cell? Water. Okay, so as more and more water enters this target cell, it swells to the point of rupture, and that's why we say cell lysis. That's one way that the complement proteins can actually eliminate a target. So you see what's going on. There's different ways you can activate it. Traditional pathway is through antibodies, opsonizing a target, saying this is a target that needs to be eliminated. But then once you activate your complement proteins, this can further help opsonize the target to make it more visible, or it can enhance inflammation, or we can form this channel called the membrane attack complex, which inserts itself into the plasma membrane of the target cell, allowing water to enter the target cell, and so therefore rupture, cell lysis. Does that make sense? Okay. All right, I think that wraps up innate immunity. Yes. I'm going to stop sharing and go back in here and pull up humoral immunity. So remote users, can you see the slides I've shared now? This is humoral immunity. Yes. Okay, perfect. All right, so I'm going to talk to you about this whole mechanism related to B cell activation. Okay, this is antibody mediated or humoral immunity. And I'll tell you why it's called humoral immunity in just a little bit. I'm going to start adding some details and we'll see how much we can get through today. But again, some parts of this I'll go ahead and revisit next class before we move any further. Okay. All right. So when you think about the adaptive response, so this is very, very specific. This is mediated by either T cells or B cells. Okay. If we are talking about B cell, B lymphocytic cell related immunity, this is called humoral immunity. Okay, because when the B cell gets activated, it transforms or differentiates into those plasma cells, which then secrete antibodies. These are proteins. These antibodies then are released and they circulate through the humors of your body, right? Any biological fluid, basically, all right? Like blood or lymph, classically considered. This is why we say this is humoral immunity, okay? Okay, but then T cells, they work a little differently. This is called cellular or cell-mediated immunity. So these don't secrete antibodies, right? I mean, they work very, very differently, a different classification of cells. So we'll go into those details next class. All right, let's go ahead and get started, and then I'll get back to the board here in just a little bit. So when you think about the adaptive response, again, this is very specific. It's specific because B cells and T cells have a very specific receptor on its surface. So there are many, many, many pathogens that you could encounter throughout your lifetime, right? And you have billions, trillions of these lymphocytes, B cells and T cells circulating through the body. Every cell has a specific receptor that it is programmed to only identify one specific foreign antigen. I can say foreign, but it's the same as saying non-self. Okay, so you'll see that interchangeably used through your lecture slides and through your textbook as well. All right, it's the same thing. Foreign antigen is the same as saying non-self antigen. Doesn't belong in your body, right? So we're mounting a very specific immune response against one specific foreign antigen. So this is going to be, again, so specific, but, and then as you can see, when we activate, for example, B cells and you got the plasma cells that secrete the antibodies can then circulate throughout your body. So it's not very localized response. You have a very systemic response here, anywhere in the body. The same can be said about the T cells. They don't secrete antibodies, but they transform into other activated cells like helper cells and cytotoxic T cells, regulatory T cells, and those cells also have a response throughout the body. Therefore, it's systemic response. But, but uh, it's a very, very sequential process, the way that this gets activated. It takes time. So there's some delays in mounting an immune response on the adaptive side of things, right? Innate response, which consists of, like, say, neutrophils and macrophages, they're not very specific, but because they're not very specific, it allows them to respond much faster too. There's not too many steps in the whole activation process. So this is good. This is a very specific response, a much stronger response, but takes time. Okay. But the other benefit to this is we will always form memory cells. Every time we activate a B cell or a T cell, we also form memory cells. So what that does is, if you had a B cell, a naive B cell that recognized a specific virus, okay, well, the majority of those B cells are differentiating into plasma cells that secrete the antibodies. The antibodies know what they need to do. They need to go after that foreign target anywhere in the body and mount an immune response to eliminate that target, right? But there is a small population of these cells. When they transform, they don't become the plasma cells, they become memory B cells. And those memory B cells just sit back and do nothing that first round. I mean, they just remember. They remember that particular virus, right? Which means that anytime later on in your lifetime, when you subsequently encounter that same target, those memory B cells can spring into action a lot faster. That's the idea. They remember that target. Okay. T cells do the same thing. Now, when neutrophils and macrophages get activated, right? I mean, they know, okay, we see a foreign target, we need to eliminate the target. They don't remember that target though, right? I mean, those they don't form memory. So that's a big difference between innate and adaptive responses. So I talked about humoral or antibody mediated immunity, which is through those B lymphocytes, and then cell or cell mediated immunity, which would be through your T cells. Okay. Okay, I've already explained this to y'all. Okay, so but I do want to add one more piece of information here. So if I had a foreign target, so when the B cell has a specific receptor that recognizes this foreign target, and then it gets activated, forming plasma cells and releasing those antibodies. How do the antibodies then work? All they can do is coat the surface of that particular target. This is opsonization, right? The one thing I did not mention is this. The B cell can only recognize a target that is extracellular. So what does that mean? Extracellular means outside of a cell. So if this virus was freely circulating in the blood, in the lymph, right, it hasn't actually entered a cell yet, right? If it was freely circulating, the B cell could recognize that as a foreign target. And if it had the right receptor, then it knows what to do with that foreign target. Does that make sense? If another B cell was passing by and it had a different B cell receptor, right? It may not know what to do with that virus. It doesn't even recognize it. Does that make sense? But if it had the right receptor, then it would recognize it. But I want you to understand, B cells will only recognize a target that is extracellular. All right, so now let's just do this and go back with my example here. I have a muscle cell. This is a normal cell, okay? Going about its business. But then what happened? The virus infected this muscle cell. You see it? So now I've got the virus here that's taken over this muscle cell. Now it's infected. You see it, right? Now the B cell, if a B cell was passing by, can it see that this muscle cell's been infected? The thing is, the virus is no longer extracellular. It's not outside of the cell. It's inside of the cell. Do you see it? Now the virus becomes an intracellular target in relation to that particular cell. Does that make sense? A B cell cannot do anything about that, right? But a T cell can. Okay, so T cells are going to go after intracellular targets like a virus infected cell, like a bacteria infected cell, right? Anything like that. Whereas your B cells can only work on extracellular targets. Does that make sense? Okay, all right. I'll come back and revisit that next class, but we're good for now. All right. Okay. So how does we talked about how antibodies are produced, and that's humoral immunity. Cell-mediated immunity, this is where your T cells get activated. They do not release, they don't form plasma cells that release those antibodies, right? Instead, they form other activated cells called, okay, I don't have it listed here, but cytotoxic T cells, helper T cells, regulatory T cells, and they are actually going to work on intracellular targets. That's why this is called cell-mediated or cellular immunity, because we're talking about something that's inside of a cell. Okay. Yeah. Virus infected cells, cancer cells, all like, yeah, cells of a foreign graft. Like, so when a transplant was not compatible, right? Okay. So I'll come back and obviously talk about cell-mediated immunity next class. So let's just go ahead and get some foundations down and see how far we can go. All right, so I'm going to use some of the schematics here, but as needed, use the board as well. If I have a normal liver cell, correct, or any cell, you'll remember how every cell has something called the glycocalyx, which is like a sugar coating on the surface of the plasma membrane. That is kind of part of the recognition of that cell. It's a signature pattern that recognizes that cell within the body. So now this liver cell has a specific glycocalyx. It's kind of similar to saying self-antigens. All right. So the liver cell is going through a whole lot of metabolic processes, right? We're processing different things. We are producing a whole bunch of different proteins. So let's say we've got a sampling of these different proteins here. That's what's expressed on its surface as what I call self-antigen. Does that make sense to you? Right? Okay, so here's the deal. If I have a normal cell, liver cell, right? Okay. Then as part of the normal metabolic processes, I've got different proteins being formed here. A sampling of what's happening inside the cell is expressed on the outside as what I call self-antigen. That makes sense to you, right? Okay, so if a B cell were to come by, and it has a very specific receptor, right? B cell receptor. The B cell should look at this going, oh, I see self-antigen. Is that right? Self-antigen means this is a normal cell. Don't touch it. Don't destroy it. Okay? Likewise, a T cell or a macrophage for that matter, or neutrophil. If it was passing by it and it looks at that liver cell and I'm saying, oh, that's self-antigen. Self belongs to me. It belongs to self. Don't touch it. Makes sense, right? But this muscle cell was infiltrated by what? By a virus. So now what do you think can happen? So initially, this muscle cell had like its own proteins being formed. Now it has the virus infecting it. Now I'm going to start making some of those blue proteins there, correct? So initially it had self-antigen, correct? But now I'm going to also present on the surface some of those viral proteins. Do you see it? So it's no longer self. This now becomes non-self, or what I call foreign antigen. Do you see it? Okay. So any normal cell is going to express on its surface self-antigen. If it's a self-antigen, don't eliminate it. It's normal, right? If your immune system was programmed correctly, your B cells, T cells, macrophages, whatever, should basically leave that alone. It's a self-antigen. But if it recognizes a non-self or a foreign antigen, that's when you need to mount an immune response against it to eliminate that particular target, right? So again, in this particular case, a B cell is not going to be able to identify this as a problem because it's an intracellular target, right? But a T cell is going to be able to, okay? Would a macrophage be able to do something about it? Absolutely, because it's non-specific. All it's doing is looking and saying, oh, that's a foreign antigen. That's a non-self antigen. What would a macrophage do? A macrophage would basically engulf that, that infected muscle cell, phagocytosis, and then eliminate it. Does that make sense to you? Okay, so self-antigen, don't touch it. Foreign antigen, yes, maybe we can do something about it, right? If it is a macrophage or a neutrophil, it's non-specific. All it's looking for is foreign antigen. But a B cell and a T cell, okay, got to think about this, right? B cell or a T cell is going to ask, I see that's a foreign antigen, but do I have the receptor to do something about it? Do I even recognize it? Because after recognition, you have to bind it, forming a complex, and that's what's going to activate the B cell or the T cell. So it's very specific. That's why I said adaptive response is very specific. All right, so let's talk a little bit more about this. So non-self or foreign antigens are basically anything that doesn't really belong in your body, which means once it's recognized by a B cell or a T cell, now you're really mobilizing the whole adaptive immune response, right? Because a self-antigen should not, a normal cell, a self-antigen should not activate an immune response, right? Because it's normal. But a foreign antigen has the ability, hopefully, right, to actually bring about an immune response because we are trying to eliminate that target, okay? So let's, went too fast. So let me talk a little bit about this. Immunogenicity and reactivity. All right, so what does this mean? Immunogenicity is where, I'm going to use the example of my B cells. When a B cell identifies its foreign target, like that virus, right? And then, okay, I said it becomes plasma cells, correct? But there's one step before that. You have one naive B cell that had the right receptor that recognized this foreign target as a foreign antigen, correct? Which then activated that naive cell, right? You got one cell. One cell cannot mount a robust immune response throughout your body. So the first thing I want to do is proliferation. Proliferation is the same as saying multiplication. So I had a naive cell, it's called a clone, right? And that cell now has been selected. It was selected because it had the right receptor that recognized that foreign antigen. Now we have to multiply that clone, and this is what we call proliferation. So we're building our army of, of recruited B cells that know what to do about that foreign target. This is proliferation. Once you have made multiple copies of that clone, those are then going to differentiate into plasma cells or memory cells. We also said this, memory B cells. Some of them become memory cells. But the majority of plasma cells that secrete the antibodies, correct? So this is our step. We've got a naive cell that was selected, which then has to multiply, and then you see differentiation into plasma cells and memory B cells, okay? All right, so what is immunogenicity? That definition tells me that a foreign antigen is able to stimulate proliferation or multiplication, right, of that specific lymphocyte. This foreign antigen, right, is able to select that clone, bringing about this proliferation process. Does that make sense? That's why I say a foreign antigen is immunogenic. A self antigen should not do that, okay? Because if a self antigen did that, then you are mounting an immune response against a normal cell. So a foreign antigen is actually immunogenic. It's able to bring about proliferation, correct? Then foreign antigen is also reactive. What does that mean? When I went through this activation process and those plasma cells would release the antibodies, correct? Then these antibodies can also then react with that foreign antigen anywhere in the body. That's called reactivity. Now, instead, if I was talking about a T cell, when the T cell gets activated, it forms, say, cytotoxic T cells. We've got, what else? Helper T cells, and we've got regulatory T cells, right? I'm gonna go with that, cytotoxic T cells. So when a foreign antigen was able to activate this T cell, it is immunogenic because it's able to bring about proliferation of that T cell, which then allows it to differentiate into those classes. But now these classes, the cytotoxic T cell, can also circulate anywhere in the body and identify that virus. That's what I mean by reactivity. Just as much as those antibodies were able to coat the surface of that virus. That's reactivity. So any foreign antigen needs to be both immunogenic as well as, okay, let's write that. and reactivity, and they need to be reactive. Both of those are qualities of foreign antigens. I'll come back to that in just a minute. All right, so let's show you, I'm going to kind of zone in a little bit more on what the antibodies are doing and go from there. So say I have a foreign antigen here. This is that red antigen. It's a foreign antigen. Every foreign antigen has different binding sites. Okay. So, okay, maybe I activated a B cell because it recognized this foreign antigen, and that first B cell released, secreted antibodies. We're going to call these antibody A. Okay. A second B cell also kind of recognized that foreign antigen kind of to a certain extent and also allowed for a different antibody to be produced. So you can see how multiple antibodies can surround or basically bind that foreign antigen. So they all have specific receptors that can recognize a foreign target, but then they can all kind of also kind of congregate on one target to make sure that that target gets eliminated, right? So these binding sites for antibodies, for example, are called antigenic determinants. So one foreign antigen could have multiple antigenic determinants, but each antigenic determinant, each binding site is only for one specific antibody. Okay, so it's very specific on the antibody side, but the foreign antigen can bind several at the same time. And really what we're doing here is it's kind of redundant, right? But it's a good kind of redundancy, just like clotting factors that you saw intrinsic pathway, extrinsic pathway, a whole bunch of redundancy built into the system, but we wanted it because we wanted to stop hemorrhage, right? So this is something similar to it. You're seeing where B cells are getting activated to target that foreign antigen. And that's not the only thing that's happening, right? The same foreign antigen is also being targeted by macrophages and what else? By neutrophils. Maybe the complement activate, the system of proteins is inserting its MAC, its channel there, trying to destroy that target. I haven't even talked about T cells, right? So there's a lot of redundancy built into the system when it comes to immunity. And this is why we don't really think about this as, oh, this is happening. Well, it doesn't quite end and then something else takes over. It's all overlapping mechanisms. It's a method. I mean, but you have to think about the delays though. Like, like there's some that are faster responding, there's others that take more time, but ultimately they all kind of converge on that target. You got to eliminate the target. Okay. And so there's multiple ways that you can do this. And I'll actually piece this together probably next class. I'll give you a scenario, we'll work through it, and we'll say, all right, let's talk about the innate system, what's happening with the B cells, what's happening with the T cells, right? And we'll talk about how they all kind of work together to eliminate the target. So lots of redundancy, okay? All right. Oh, fun times. MHC proteins. Okay, so this is not so quite relevant for B cells, but I need you to understand this because when I get to T cells, it's very, very, very relevant. All right, gotta change my angle here. There. Okay, in next class, I'll go into more details, but I am going to talk to you about a population of cells called antigen-presenting cells. Um, Antigen-presenting cell APCs are of three different varieties. Your classical APCs are something called dendritic cells. Does that ring a bell? In ANP1, when we discussed the integument, we talked about these cells. These are dendritic cells. These are the policemen that were patrolling like the epidermis and maybe the lower dermis regions. And all they're doing is they're sounding the alarm if they identify a foreign antigen. If a dendritic cell was patrolling, right, and we came across this normal liver cell, what is the normal liver cell expressing? Self-antigen. Dendritic cell says you're good, right? We don't nothing to worry about here. But when that dendritic cell comes across this infected muscle cell, it says, oh, wait a second, that's not self-antigen anymore. That's foreign antigen, right? And so what happens then is it basically takes fragments of this and expresses it on the surface of that dendritic cell, saying, look what I found. I have this foreign target that I've identified. I'm not the right person for the job. I don't know what to do with it, but I want all of your specialized cells that know what to do with it, y'all need to go after the cell and eliminate it. That's why it's called an antigen presenting cell. It doesn't destroy the target. It just presents antigen. Make sense? So everybody else that are looking at that antigen, no. If it's self-antigen, don't worry about it. If it's foreign antigen, oh, wait, maybe I'm the right person for the job. Does that make sense to y'all? So that's an antigen presenting cell. So that's my true class of APCs. But I'm also going to show you later on how B lymphocytes can also be antigen presenting cells. It's a self-serving purpose now. And then, you know, your innate response, non-specific, when we talked about macrophages, yeah, macrophages are also antigen presenting cells. Okay, so I'll come back and explain that next class. But I want to show you what this means. So let's say I have a dendritic cell. It's a normal cell, right? It's part of the immune system. It's a normal cell. And it's basically got its own thing going on. It's got its own proteins going on, right? It's going to express that, and it's something like that. It's called major histocompatibility complex protein, MHC, okay? There's two classes of it. I'm not going into that detail today, but that's coming up next class. That's an MHC protein. How did this come about? This is actually part of what's going on inside that cell. Does that make sense? This is some of those proteins being expressed there on the surface. It's called MHC protein. In other words, this belongs to that dendritic cell, correct? MHC proteins are self-antigen. They are self-antigens to that particular dendritic cell. All right. But what the purpose of this MHC is that it's a placeholder, right? It's like a platter. I'm serving something on my surface, and I want everybody else passing by to see what I'm presenting. Antigen presenting cells. You see that? I'm presenting something on that MHC. So if this dendritic cell, it has its MHC protein on its surface. When it passes by and it sees that liver cell, it's normal, it's self-antigen, right? It's going to sample that and go, all right, self-antigen, I'm just going to express self-antigen from that liver cell, correct? If a macrophage comes by, if a B cell comes by, if a T cell comes by, right, it's going, well, mostly not a B cell, T cell. If a T cell comes by, it says, okay, I recognize MHC protein, and what are you presenting on that MHC protein? This is self-antigen from a normal cell in the body. Don't do anything about it, okay? But what if that dendritic cell came across that infected muscle cell, right? Then it's saying, oh, wait, that's an infected cell. I see it's expressing foreign antigen. So now what happens? Now I'm expressing, not self-antigen, I'm expressing some of those viral proteins there as well. So this becomes non-self-antigen. You see that? Now a T cell goes, oh, I recognize this is MHC protein. What are we expressing on this platform? Oh, that is foreign antigen, right? Then the T cell might say, okay, I see it, but I don't have the right receptor. I can't do anything about it. Or maybe a different T cell clone comes by and says, wait, I see MHC, and I also see that it's a foreign antigen being expressed. Oh, I do have the right receptor. I know what to do about that, right? And that would activate that particular T cell clone. You see what I'm saying? Hold on to that thought, just 1 minute. But do you understand the overall idea here, right? So a dendritic cell is an antigen-presenting cell. It has on its surface self-antigens, which we call MHC, major histocompatibility complex protein. That's a mouthful, isn't it? MHC proteins. But the idea behind those MHC proteins is that it's actually presenting some kind of antigen. It could be self-antigen. If it's self-antigen, your immune cells should not touch it, right? But if it's a foreign antigen that's being presented, then again, is this the right B cell or a T cell to do the right job? Okay, back to your question. Oh, I'm about to explain all that fun stuff next class. You got to hold on to that thought. I'm going to show you different pathways. Yes, answer to that is yes, but I'll show you how that works. Okay, does that make sense to you all? Good. All right, so what I'm going to come back and talk about MHC proteins next class. It's not so relevant today for B cell activation, but certainly for T cells. All right. So if a T cell was passing by, right, a T cell, say a T cell was coming across this liver cell, which has, it's a normal cell, self antigen. The T cell cannot even see that self antigen. It cannot recognize that self antigen. Can the T cell recognize this as foreign antigen? No, it cannot recognize any antigen unless the antigen is being expressed on an MHC protein. Do you see it? This is why those dendritic cells are very, very important for presentation of an antigen to a T cell. B cell, nah, no, we don't quite need this. A B cell can see, oh, that's self antigen, this is foreign antigen, I know what to do with it. But then why are they here? I'll explain that next class. They really just need some help. That's it. Okay. But T cells are extremely dependent on antigen presenting cells such as dendritic cells to present the antigen to them, to present the self antigen so we can walk away, or to present the foreign antigen so we can get activated. Okay. All right. I'll come back and discuss that more next class. Like I need to close some markers here. All right. So when you're thinking about the adaptive response, we have your B cells, you have your T cells, you also have your antigen presenting cells. So I'm going to go ahead and start talking a little bit about this whole maturation process or development process. Whether it's B or T cell, the process is pretty similar, okay? So there's some variations, obviously. So here are your overall steps related to this activation process. In your first exam, and again, if you've taken ANP1 with us, so it's the same idea, really. Your multiple choice exams have a lot of word-based questions, right? I mean, but then you also have some schematic-based questions. You saw some of those in your first exam. You're going to continue to see schematic-based questions throughout the semester. So for your next exam, oh yeah, this schematic might show up, just saying, okay? Because this one's a good one to kind of talk about the whole developmental concepts of your B cells and your T cells. So please kind of be familiar with this. So let's spend some time here. So the first thing is, all right, I'm keeping it generic right now, the B cells and T cells. Where do they develop? Where do they come from? They come from the red bone marrow, from a common progenitor cell called hematopoietic stem cells. We talked about this in the blood chapter, right? Okay, starting from those stem cells, depending on what cues they receive, right, they might commit to become, all right, B cell versus T cell. Remember how they originate from the lymphoid lineage, right? All right, they become these precursors for your B and your T cells. So then they have to go through this process called maturation. It's an education process, okay? This part can get a little complicated, so I'll come back and spend some time here. But maturation of your B cells will happen in the red bone marrow. So if I have a B cell precursor, it stays right there to go through the maturation process. But if I have a T cell precursor, it's going to leave the red bone marrow, migrate to the thymus. You see that? And the thymus is where T cells will mature. Okay. Once these cells have matured, when you have a B and a T cell that has matured, this is where we call them naive cells, right? Naive B cell, naive T cell. What does that look like? Any B or T cell that's naive, that's properly educated, either in the red bone marrow or in the thymus, will have a specific receptor. Do you see that? It's going to have the B cell receptor or your T cell receptor. So what does the receptor do then? The receptor is then able to identify one foreign antigen. Okay, is that good enough? Nope, not good enough. Once I recognize the foreign antigen, I also have to bind the foreign antigen, forming a complex between the foreign antigen and the receptor. That's what's going to activate that B cell or T cell. Does that make sense to y'all? Okay, so we're going to talk about as the lymphocytes mature, they come out of that process with two properties. They are immunocompetent and they are self-tolerant. And I might have time to talk a little bit about that. Okay, so as we have immunocompetent and self-tolerant, T cells and B cells that then leave wherever they went through education, and then they start to circulate through the blood and the lymph. You're seeing a lymph node right there, okay? So this is what I call seeding of secondary lymphoid organs. So what is a primary lymphoid organ? A primary lymphoid organ is where that education was occurring. So it's the red bone marrow for the B cells and the thymus for the T cells, right? These are your primary lymphoid organs. Once they leave the primary organs, then they start moving through the vasculature and the lymph nodes. We call this secondary lymphoid organs. Okay, this is where they're circulating. And more than likely, it's in those lymph nodes that your naive B and T cells will encounter foreign pathogen, right? So when it encounters foreign pathogen, so what do I have here? I have a B cell that's been properly programmed. You see all of those Y-shaped receptors on its surface, right? So it's got the right receptor. It's been properly programmed, and they are both immunocompetent and self-tolerant. And now as I'm passing through this lymph node, I'm encountering this red foreign antigen. Do you see it? Correct? So now the B cell says, all right, I see that this is foreign antigen. Do I have the right receptor? If I have the right receptor, then what? The receptor will bind the foreign antigen. You'll see that happening here, forming a complex with the foreign antigen, which then activates this B cell. Once you activate the B cell, the first thing that happens is proliferation, multiplication of those clones, which then allows it to differentiate into very specialized cells, your plasma cells and your memory B cells, majority being plasma. Plasma cells are then going to secrete the antibodies, and then those antibodies can go find that red foreign antigen anywhere in the body. All right. This is the whole development and activation up all the way up to activation of your B cells. T cells, very similar, except I have to come back and talk to you all about what the MHC proteins does and how it helps with that activation process. Okay. So this is the overall idea. And I want to talk to you a little bit about maturation here. Maturation really encompasses two things, immunocompetence and self-tolerance. I'm going to go ahead and get a head start and explain it to you. But then I know y'all are not going to fully follow it. So when I get back next class, which is what, next week, we will come back here and start from here, redo it again, and then go forward. Okay. Okay, so for immunocompetence, what does this, you got to understand these concepts. Immunocompetence, so basically this is the maturation or education process that's occurring for the B cells. This is occurring in the red bone marrow. For your T cells, it's happening in the thymus, correct? So when a B and a T cell completes their education process, they have two qualities. They have two things that they are capable of doing, right? One is immunocompetence. What does that mean? Immunocompetence tells me that a lymphocyte, whether it's B or T, a lymphocyte can recognize one specific non-self or foreign antigen. Notice it didn't say self antigen, right? A B cell or a T cell should not recognize a self antigen, and it should certainly not bind to it. Does that make sense, right? If a B cell came across this normal liver cell that was expressing self antigen on its surface, if the B cell by mistake recognized this and bound that self antigen, it would mount an immune response against that normal cell and eliminate a normal cell. Okay. Okay. This is autoimmune disorders. Okay, this is kind of the premise behind autoimmune disorders. We don't want that. What should an immunocompetent B or T cell be able to do? They need to recognize one specific foreign antigen and they need to bind it. Okay, right? Not self-antigen. What else should they do? They should be self-tolerant, which is exactly what I explained. If it recognizes a self-antigen, they should be tolerant towards that cell. They should not try to attack that cell. Does that make sense? So every B and T cell every B and T cell, or when they go through that whole education or maturation process, they need to be immunocompetent. So immunocompetent is where they can, oh, went too fast. They need to recognize It's not like that. I can spell this. Again, they need to recognize only one specific foreign antigen. And they need to bind it, forming a complex. That is immunocompetence. Second is self-tolerance. What does that mean? It means do not bind self-antigen. That makes sense, Jill? Yeah, okay. How is a B or T cell immunocompetent? How is it able to recognize one specific foreign antigen? Because it has a receptor, right? Because it has that receptor that can recognize that foreign antigen and then bind it. That's immunocompetence. But that same receptor should not bind a self-antigen. It should be self-tolerant. Does that make sense to y'all? Okay, I'm going to use my next slide, which can look a little confusing, but I'm going to go ahead and work through that schematic. But again, like I said, next class, we'll start back here. We'll map it out on the board again before we move any further, okay? Are we good with this? Do you all understand this? This is the maturation process that's occurring. This is a tricky concept. You may, before you come in next class, certainly go ahead and review the video and make sure you're kind of absorbing it, and then we'll map it out again, okay? So every B and T cell has to, if it's properly educated, if it's been properly programmed, then it's going to, T cells are going to leave the thymus, B cells are going to leave the red bone marrow, exhibiting these two properties, right? They have to be immunocompetent and they also have to be self-tolerant. So the next slide actually explains this in relation to T cells. Remember I said T cells, okay, remember what is my goal here? I need to be able to recognize a foreign antigen, right? And I should not bind a self-antigen. But remember I said T cells, they cannot even recognize self or foreign or anything like that unless those antigens were expressed on MHC protein. So what's explained here is in relation to T cells, their maturation process. Okay, so I'm going to explain it to you. So it consists of two steps here. Again, don't worry, I'll map it out again on the board, right? And we'll figure this out one more time. The first is a positive selection step. The second is a negative selection step. So you're seeing two cells here. Okay, I'm going to start at the top. I've got in blue here on the right, this is a developing T cell. So which means it's developing or maturing in the thymus, right? So this is happening in the thymus. I have a developing T cell, got really quiet. Okay. And here on the left here, I have a purple cell, which is one of your dendritic cells, your antigen presenting cell, right? So I have my antigen presenting cell, and this structure here on the surface of the antigen presenting cell, that is my MHC, self-MHC. Do you see it? Okay, now what happens here? If this is a developing T cell, the first thing that T cell needs to do is recognize MHC, correct? So if you're looking at the overall structure of this purple region versus the blue region, you see how they kind of mismatch? They don't quite fit together, which is basically saying that this T cell here is not able to recognize that MHC. Do you see it? If I cannot even recognize the MHC, I'm going to throw out that cell. I mean, that's not going to be able to move forward. Does that make sense to you? So unfortunately, in this first scenario on the top, this developing T cell was not even able to recognize self-MHC. So what are we doing to this T cell? It's not good enough. We're going to destroy it. Does that make sense to you? Right? Now on the bottom here, you actually see a T cell. You see how it matches up nicely with the purple portion on the left, which means I'm saying that this developing T cell was able to successfully recognize self-MHC. Do you see it? That's why this is positive selection. Yes, positively, I was able to recognize self-MHC. I can put a check mark against it. Is that right? If I'm able to do that, this cell moves on to step 2. Do you see it? Okay, this is a T cell. The first thing that developing T cell needs to do is a positive selection step. I need to positively be able to identify self-MHC. Is that right? And then I can move on. Okay, I've moved on. Here's my next step. Next step is a negative selection step. Okay, so here's what's happening. So then the T cell that's developing says, okay, I recognize MHC. I see it, right? I mean, I understand it's MHC. I got it. What does an MHC protein do? It's a platter. It's a platform that presents some kind of an antigen, isn't it? Right? So if I go back to my schematic here, you see this red antigen right there? Correct? So that red antigen is what's being presented by the MHC, but that happens to be a self-antigen. And what did this developing T cell do? In the first scenario here, developing T cell says, okay, I recognize MHC, but then it bound self-antigen, normal antigen. Does that make sense to you? In other words, this is not tolerant towards self, right? Which means that this developing T cell should not proceed. It needs to be eliminated. So in the bottom scenario here, uh, pointer, developing T cell is able to recognize the self MHC, but you see that there's a gap there, meaning it did not bind the self antigen being presented on that MHC. This is good, right? Because it needs to be self-tolerant. And you see why this is called negative selection? Negative selection means do not bind self antigen. That makes sense to you? So if I can say, as the T cell is maturing in the thymus, if I can do two things, the first step is positively identify MHC, positive selection, check mark, right? Move to the next step. Then in the next step, I am not binding self-antigen. That's why negative selection, correct? If I can put check marks against both, then this is a properly programmed T cell. It's properly matured. It is both immunocompetent and it is self-tolerant. That makes sense, Jill, right? So this needs to happen in the thymus if you're talking about programming or maturation of your T cells. Okay, is that somewhat? I mean, do you understand the basics maybe of it? We'll come back and do this next class. So what this is really important. This will kind of set the stage for what we need to talk about with T cell immunity as well. Okay, so I'll let you go. Do well on your exam. For those of you taking exams today, and please read ahead, watch the video if you can, or review the slides, come prepared. Gonna pick up the pace a little bit, okay? Remote users, does anybody have any questions for me? If not, I'm gonna say bye. Okay, have a wonderful rest of your day. Bye.